Today is the 19th November 2020, and we have our first presenter from Israel. So it's my really big pleasure to welcome Neta Regev Rudzki. And Neta's lab is studying cell communication and signaling pathways of the deadly human malaria parasites, the Plasmodium falciparum. Specifically, her lab is focusing on investigating the role of parasite-derived extracellular vesicles in reshaping different host targets, how the parasite deliver beneficial messages from a distance while growing inside its host, the red blood cells. So her talk today is titled, How Malaria Parasite Tricks Its Host. Please welcome Neta.
Thank you. Thank you, Carolina and Jan, for initiating this forum to keep our community alive, even if this crazy times. And thank you for inviting me. Okay, so malaria. Despite many years of research, malaria is still one of the most devastating infectious diseases in humans. It kills 1,000 children every day, children under the age of 5 years old. The drug resistance of the parasite is a major problem, and so far, 2020, there is no effective vaccine available. Plasmodium falciparum is one of the species of Plasmodium that causes the disease in humans, and the life cycle of the parasite is fascinatingly complex. The parasite develops ways to survive in two different biological environments, two different hosts, mosquitoes and humans. Infection starts when an infected mosquito releases parasite forms into the skin. These parasite forms travel to the liver, enter the hepatocytes, and divide like 10 days. Then thousands of parasites are released into the bloodstream, enter another host cell, the red blood cell, and start the blood stage cycle. This is the stage that causes the disease in humans. This is the stage that kills the children. In 48 hours, the parasite invade the red blood cell, develop and divide to daughter cells. Some of these parasites are differentiated into the sexual forms, male and female gametocytes. These are the forms that can be taken up by a female mosquito during a blood meal, and then these sexual forms can mate in the mosquito's midgut. The decision to commit to the sexual stage is once in a life cycle decision, and so far the molecular mechanisms behind this decision are still mostly unknown. While it is clear that for such a complex journey, the parasite must develop means to sense the host, communicate, and coordinate their actions, these means remain one of the greatest mysteries in malaria. Just after invasion into the red blood cell, the parasite is encased by two separate membranes. And the parasite exports hundreds of proteins into the host cytosol in order to remodel it. And how can we detect these parasites? Simply by Giemsa smears, as you can see here. This is an infected red blood cell, and this is uninfected red blood cell. So these parasites are intracellular parasites. They live inside the red blood cell. And indeed, in the lab, we feed them every second day with human blood that we get from the hospital. Previously, we provided the first evidence that these parasites, despite all the membranes and the barriers between them, are able to exchange active cargo, episomal plasmids between them. We then found how the parasites communicate via secretion of extracellular vesicles. Now, it is true that in order to understand the function of the vesicles in signaling in malaria, it's essential to purify a large number of these vesicles and determine their cargo. And this is exactly what we are doing currently in the lab. And even at that early stage of the analysis, we already found that the parasite used these tiny packages to release remarkable components, including parasites and host proteins, and parasite and host RNA and lipids, and even parasitic genomic DNA protected within these vesicles. And the data that received from this analysis serve as the firm base for our published studies and unpublished studies. So today I would like to share with you two independent studies in this line of research. But before we move on, let's just look at the big picture for a moment. We first showed that these intracellular parasites, despite the barriers, are able to communicate, to share information. We then found how, via secretion of extracellular vesicles. We could also say something about why the parasite established this communication machinery, and I didn't show you the data, to promote their sexual commitment. We then move on to analyze the vesicle cargo components, and I told you about the DNA and RNA and so on. But when we think about it, these parasites have to face very hostile environment in their super crazy life cycle. So we can imagine that a parasite may use these armed vesicles not just to talk between them, but also to alter, even manipulate their host response. And if that is the case, one of the main targets would be the host immune response, or even their most direct and essential host cells, the red blood cells. So these are the two studies that I would like to present today. So we developed several EV methods, including the one that is based on image stream flow cytometry, where you can, using this method, you can monitor the uptake of the vesicles into different target cells. And the advantage of the image stream flow cytometry is that it can really give you robust data, as you can screen more than 50,000 cells at once. And indeed, using this image stream flow cytometry, we were able to show that the parasitic vesicles can be taken up by host immune cells as monocytes. And then the DNA cargo is released inside the cytosol and is recognized by the STING pathway. And we were able to dissect step by step the signaling pathway to induce type 1 interferon response, most probably as a decoy mechanism to confuse the immune response while the parasites are growing in a distance inside the red blood cell. Now, when we look at the type 1 interferon response, we measure two parameters, as you normally do. We look at the RNA level of these cytokines and chemokines of type 1 interferon response when we introduce the vesicles, and we look at the protein, the secretion level of these cytokines and chemokines. So when we look at the RNA level, everything worked perfectly. We introduced the vesicles to monocyte, we got high levels of the transcript of these cytokines and chemokines. But when we look at the secretion level, one chemokine was misbehaving, and this is IP10 or CXCL10. No matter how many times we repeated the experiments, we could never detect the secreted protein chemokine outside of monocytes. High level of its RNA, but no secretion, as opposed to the positive control. So to be honest, at that stage, I thought, well, one chemokine is misbehaving. I wasn't getting really excited by these results. But in fact, my postdoc from the other side, she thought that this is really exciting because maybe there is something there within the vesicles that block the secretion specifically of IP10. And when she looked at the literature, what is known about IP10 and malaria, here it started to be really interesting, as according to several studies coming from endemic areas of malaria, high levels of IP10 in the blood of patients associates with the severe cases of malaria, cerebral malaria. So these are the cases where the parasite gets to the brain, stuck in the BBB, that leads to coma and death. And apparently, high level of IP10 associate with these cases. So IP10 is the marker for cerebral malaria. And surprising enough, in the non-complicated malaria cases, IP10 levels in the blood of the patient is relatively low. So obviously at that stage, I thought, well, this is important enough, let's keep going. So first we confirmed that indeed we get the same effect when we use primary monocytes that we got from healthy donors from the hospital. And again, we introduced the parasitic vesicles, no secretion of IP10. But then what we found that actually it's not that there is no secretion, that the protein cannot be secreted in monocytes, but actually there is complete depletion of IP10 within the monocytes when we introduce the vesicles. So no expression of IP10 at all when we introduce the vesicles, as you can see here by Western blot or by ELISA assay. And from that moment, we started to systematically investigate the molecular mechanism underlying IP10 depletion. And we took many different directions, and I'm not going to present them, all of them today, but just some. So for instance, we asked, maybe it's related to the stability of the RNA. The vesicles coming, something happens, and IP10 mRNA is not stable enough, and that's why we cannot detect any protein. So we tested that, and that turned out not to be the case. So the RNA is relatively stable. Then we thought, okay, the RNA is relatively stable, but maybe there is something else that blocks the export of the transcripts from the nucleus to the cytosol, and that's why we don't get to see any protein. There's no translation. So we tested this phenomena is termed the nuclear retained mRNA, and we tested the maturation of the mRNA, and again, this turned out not to be the case. So the mRNA is relatively stable and it gets exported to the cytosol. Then we thought, okay, so maybe when the vesicles come in, something blocks the association of the ribosomes onto the transcript of IP10, and that's why we cannot detect the protein. There is no translation. So to assess the association of the ribosomes with IP10 mRNA, we established polysome fractionation assay. And in this assay, you get several fractions of the transcripts, starting from the free RNA, so there is no translation for this transcript, there is no association of ribosomes on these transcripts, to the heavily translated polysome fraction, where there are many ribosomes loaded onto this mRNA in this fraction. So when we look at another cytokine, for instance, interferon beta, when we introduce the parasitic vesicles or in the positive control, we got a heavy translation, polysome fraction for this mRNA loaded with many ribosomes. But what happens to IP10? In the positive control, heavy translation in the polysome fraction. But when we introduce the parasitic vesicles, we got this massive shift into the free RNA fraction. So no association of the ribosomes to this specific RNA, only when we introduce the parasitic vesicles. And at that stage, we kind of got stuck. So how do we continue from here to investigate the molecular mechanism behind this regulation? So what do we know about our system? We know that STING is activated, I told you from the previous study. We know that there is internalization of parasitic vesicles carrying genetic material. And we know that there is translation inhibition for IP10. So then we look in the literature if there's any other similar systems that combine these three elements, these three principles. And there was a paper that came out 2 years ago from Jonathan Kagan's lab. And what they showed there, that in the cases of RNA virus infection, that activates the RNA sensor RIG-I, and then and that leads to activation of STING. And together, that leads to restriction of the translation of the viral and the host target proteins. So combining these elements. So given this data, we decided to examine if RIG-I, the RNA sensor, is also involved in our system. To address that, we used RIG-I knockout monocytes that were generated by the CRISPR-Cas9 system. And as you can clearly see here in the RIG-I knockout monocytes, all of a sudden we got expression of IP10, and we could detect it by a Western blot and by ELISA assay. So the translation inhibition doesn't work anymore. That means that RIG-I is a key player in our system. Now, 2 more points that I would like to mention when we talk about translation inhibition mechanisms. So the first one is that these mechanisms are sort of divided into 2 main modes, the global translation inhibition and the sequence-specific translation inhibition. And from our ribosomal profiling, we know that in our system, there is no global translation inhibition in monocytes when we introduce the vesicles because we got similar profiles between the treated and the untreated cells. So then most probably there is sort of a certain sequence, certain region within the RNA of IP10 that carries this information that controls the translation inhibition. And the second point that I would like to mention is that AU-rich elements located within RNAs are known for their ability to fine-tune mRNA translation and stability. And indeed, when you look at the 3' UTR of the IP10 transcripts, we could identify several AU elements in this region. So to examine if this region controls the translation inhibition and carry the information for this translation inhibition, we built this construct where we fuse this region to a reporter marker. So the reporter marker is always expressed within monocytes unless there is something there that controls its translation and reduces its translation. And this is what we found. When for the fusion protein, we could see a complete depletion of the reporter protein. So there was no translation of this for this construct. We then decided to zoom into this area and divided it into 3 more subdomains and examine the effect of each of these domains on the reporter marker and to see to test its translation. And what we found that in this small region of almost 90 base pairs that controls the translation inhibition. So all the information is in this area, in this sequence. So if this is the region that contains these AU elements and somehow blocks the association of the ribosomes, that means that there must be a protein that binds to this element, and that's why it blocks the ribosomes. But how can we find this RNA binding protein? So we knew that this protein must tick several boxes. It must be RNA binding protein, must bind to AU motif. It needs to be involved in translation regulation, and even better if it is involved in immune responses. But still, there were way too many candidates that can fit to this role, and we were kind of, again, getting stuck. And about the same time that we were seeking for this candidate protein, I was invited to give a talk in the annual EV meeting in Holland and presented this data. And then Esther approached me saying that they're actually currently working on a protein that maybe has a similar role or maybe can fit and tick all these boxes and play the role in our system as well. And this is HUR. And to be honest, till the moment that Esther mentioned this protein, I never heard about HUR before. But indeed, it ticks all the boxes. So HUR is involved in post-transcriptional regulation of many genes, and in particular of cytokines and chemokines. So at that stage, I thought, well, let's just give it a go and see. And to address that, we silenced HUR using the siRNA assay. And as you can see here, under silencing of HUR, all of a sudden we got expression of IP10. So, so no more translation inhibition. So we were very excited with this result and decided to take it even one step further and examine if indeed HUR directly binds to the RNA of IP10. And to address that, we had to establish this native RNA immunoprecipitation assay, where you come with an antibody to pull down HUR, and then you examine which RNAs are associated with the pull down sample. And the results were really striking. As you could see here, we got really high levels of IP10 mRNA coming in the pull down of HUR, but only when we introduced the parasitic vesicles to monocytes. So if I sum up what I showed you so far, we found that these secreted vesicles can be taken up by monocytes, and then the RNA cargo in this regulation is recognized by the RNA sensor RIG-I. That leads to a chain of events, and eventually it leads to the recruitment of another RNA binding protein, HUR, to a small AU elements region in the 3' UTR of the mRNA, and by that it blocks the association of the ribosomes, and there is no translation of IP10. But why would the parasite invest all these efforts just to stop the translation of this chemokine? And keep in mind that this chemokine is a marker in many cases for the severe cases of malaria, cerebral malaria. So these are the cases where the immune system is out of control and the host circumstances are really bad. But they are really bad also for the parasites. And the parasite must move on. The parasite must grow faster, develop faster to the sexual forms, and move on to the mosquito vector. They have to change their tactics and plan their escape actions. So maybe IP10 itself serves as an alert facilitator back to the parasite to alert the parasite how bad the host circumstances are. And this is the time to move on and change our tactics. And this is exactly what we found. When we introduce IP10 to the parasite in many different conditions and only the chemokine IP10, we got higher level of parasitemia. So somehow the parasite can sense its presence. And we could detect these beautiful male and female parasites coming up only when we introduce this chemokine. And as you can see here, within the first 24 hours, when the parasite can sense IP10 in its environment, we got higher activation of the sexual genes of the parasites. And just to remind you that these sexual forms are responsible for the transmission of malaria from the human host to the mosquito vector. So if we know how to block these sexual forms, there will be no more transmission of malaria. So we propose this two-phase model mechanism of IP10 regulation, which returned a decision sensing system. So the parasite works really hard to reduce IP10, and that's at the early stage of the infection or the non-complicated infection. But when the parasite can no longer reduce IP10's level, and this is at the later stages or the severe cases, and IP10 is then secreted, the parasite can sense it and change its tactics, go faster, and develop to the sexual forms to move on to the mosquito vector. So that was about the host immune cells. But what about the most direct and essential host cells, the red blood cells? So this study was performed in a very successful collaboration with Michal Sharon from our department, experts in the proteasome biology. But before we move on, let me just remind you what makes the red blood cells such unique host cells. So these cells have to survive something like 4 months, but they have already lost their internal organelles. So there's no nucleus, no mitochondria, no ER, no synthesis of new proteins. And that means that these cells rely exclusively on the existing pool of proteins, and that led the red blood cells to develop unique mechanisms of cell survival and cell death. And if we talk about pathogens reshaping these sophisticated cells by vesicles, then most probably the DNA and the RNA cargo won't affect them as much, but rather the protein cargo. So let's just keep that in mind. And since the parasite invade the red blood cells every 48 hours, our mode of work was that we harvested vesicles from the infected red blood cells, introduced them to naive, healthy red blood cells, and then examine different molecular traits of these treated cells. And in this study, we combined biophysical, biochemical, omics, and computational methods. So the first question we ask, what happens in terms of parasitemia levels when we introduce the parasites to these treated cells? And here we got higher level of parasitemia when we introduce them to the treated cells, suggesting that a parasite use these vesicles to promote their growth before they invade the red blood cells, before they invade this host. Now, it is known from other studies that it is much easier for the parasite to invade red blood cells that the membrane is softer. We used atomic force microscopy to measure the stiffness of the membrane. And as you could see here, we got a much softer red blood cell membrane when we introduce the red cells to the parasitic vesicles as opposed to the control vesicles coming from uninfected red blood cells. So the membrane becomes softer. And the mechanical properties of the red cell membrane are determined by the underlying cytoskeleton network. So we decided to take an advantage of this AFM method and image the cytoskeleton for the treated cells. And we got this massive disruption in the cytoskeleton. Either we could see some phenotypes of holes or sort of meshing and melting of the cytoskeleton filaments in the cases of the parasitic vesicles, as opposed to the uninfected derived vesicles. So far, I was talking about 3 different phenotypes: the growth elevation when we introduced the parasitic vesicles, the stiffness, we could detect membrane which is much softer, and this cytoskeleton disruption. And the question is if we can identify what is the main component that is carried via the vesicles and responsible for these 3 phenotypes. And as I told you before, most probably the protein cargo would be more relevant to this kind of study rather than the DNA and RNA because of these sophisticated cells. So we performed proteomic analysis to the parasitic vesicles, and 2 subfamilies drew our attention. We could detect large group of host and parasite kinases, but I'm not going to talk about this direction today. But we could also see many host and parasites proteasome subunits protected within these vesicles. And I mentioned these results to Michal from our department over a coffee break, and she decided to join us and collaborate on this direction. So obviously we had to show that these proteasome subunits are indeed in the vesicle function using optiprep gradient. But then we had to transfer all these complicated biochemical assays for the proteasome and to move them into the EV field, so to study their presence in the vesicles. And the first question we asked whether these subunits that we could detect by the proteomics of the proteasome, maybe they are not just there, but they are in fact assembled to give an intact active and functional proteasome, like completely assembled proteasome, 20S proteasome within the vesicles. So using native gel, in-gel activity assay, and many other biochemical assays that I'm not going to present them today of the 20S proteasome, we indeed found that the parasites exports an intact and assembled and highly active 20S complex encapsulated within these vesicles. And to the best of our knowledge, this is the first evidence for the shuttling of this 20S proteasome via the parasitic vesicles. And I think it might be more of a general mechanism that is employed by other systems, EV system, and not just malaria. Because we could see in the literature that there are many examples of other, proteomic data. And if you look at from cancer and many different systems, you can see that many of the proteasome subunits are indeed coming out of this analysis. So then we ask, as I said, we could identify these 3 different distinct phenotypes. And then we ask if the exported 20S complex is actually involved in these phenotypes or is responsible for these phenotypes. So to address that, we had to repeat all our experimental approaches, but this time using proteasome inhibitor. And we used several proteasome inhibitors in each of these experiments, and we got similar results. So let's start first with the parasitemia level. So yes, when we introduced the parasites with vesicles that were treated with the proteasome inhibitor, we could no longer see this growth advantage when we introduce only the parasitic vesicles. Same with the stiffness of the membrane, as you can see here. This time when we use the proteasome inhibitor, we got similar levels of young modulus, similar to the non-treated cells. What about the cytoskeleton disruption? So here again, we could see more of the wild type phenotype of these cells rather than the disruption that we can see with the parasitic vesicles when we introduce the proteasome inhibitor. But in order to fully quantify these results in an unbiased way, we decided to take these results one step further and established AFM machine learning analysis, where we ask the machine to actually, in an unbiased way, screen all these hundreds of AFM images and tell us what are these, from all these samples, which are the most similar one. And we could see that it was really nice to see from this unbiased method that indeed, when we introduced the proteasome inhibitor, we got similar phenotype to the non-treated cells, very similar values as opposed to the parasitic treated red blood cells. So the proteasome is involved in these 3 phenotypes, in these 3 roles. But as you know, proteasome, this complex degrades proteins, right? So this is the function of the complex. So we decided to take these results one step further and examine whether we would be able to identify the target proteins for degradation by this exported complex. And the answer is yes. And I'm not going to show you again all the data, but what we found that all of these cytoskeleton proteins are the targets of the exported 20S complex. And all of them belongs to the same apparatus. And we use several biochemical techniques to show that indeed this exported complex degrades this protein, the matin, adducing, anchoring, and so on. So just to sum up this study, we showed that the parasites secrete vesicles that carry the unassembled and active 20S complex. And this complex is involved in modifying the mechanical properties of the host red blood cells and degrading cytoskeleton proteins. And by that, assist the parasites for its invasion and for its growth. And I must say that during the past 5-6 years, there are more and more examples of other parasites that produce vesicles to facilitate their growth and modifying different target host cells. And just to end, I truly believe that to fight malaria, we have to understand the basic cellular biology of this deadly pathogen. And with that, I would like to thank all the lab members and our collaborators in Israel and outside of the country. And thank you.
Thank you so much, Neta. So for everyone now, please post your questions into the chat box, and then we will unmute you and allow you to ask the question live. I think you can stop the sharing, Neta, right now.
Yeah.
So there's clearly a lot of complexity here in the communication between parasites and the host. So let's see if Hernando del Portillo have no question. Two questions. Go ahead, shoot. I don't know what the questions are, but that's fine.
I was typing them, Jean. But
yes, shoot.
It was too fast, you know. Neta, thank you so much for the talk. It was absolutely excellent. I have two questions, one on the first part of the talk and the second one on the second one. The first one is, Andy Waters many years ago demonstrated that there was an enormous amount of translational inhibition of several, you know, proteins in malaria just before gametocytogenesis entering in the mosquito. So I am amazed about the specificity of the IP10 inhibition. Have you checked if, you know, this mechanism also affects other genes that will be, you know, released for translation in the mosquitoes?
Yeah, this is an excellent question, Hernando. This is something that we are currently doing, looking for other candidate genes. I would guess that there are others. It's not just this IP10. Probably others, maybe that they have similar, you know, sequence or similar function. We don't know, but this is something that we are looking into it now. So we are examining several other target proteins. If you have some that you can recommend us, suggest us, send us the list, and we'll test them. This is something that we are doing right now. Good question.
And the second question is a little bit more tricky, because to what extent the observations that you're seeing with EVs, you know, and red blood cells is just an artifact of in vitro culture? I'm asking you this because during erythropoiesis, red blood cells, you know, release, you know, most of the adhesins molecules in order to, you know, fulfill the oxygen CO2, you know, interchange through the whole body, so they can really move without attaching. So if you put the extracellular vesicles back to the red blood cells and you alter these red blood cells will be target for spleen destruction. So I really would like you to comment, you know, if this mechanism will really be seen in vivo, because again, during erythropoiesis, the red blood cell releases, you know, through EVs all of these adhesins. So I would like you to comment on that.
Yeah, so the thing is, I think for me, what really convinced me that this is like, we can see something like really specific here is that when we use these inhibitors for the proteasome, we could no longer see it. So if there are any proteins that are there or something else that is, you know, affecting it like in a non-relevant way, then we would expect to see it. And we tried several proteasome inhibitor, and we could no longer see this growth elevation or this phenotype of the mechanical properties of the red blood cell. And even if you just take uninfected vesicles, if you say, oh, just it's the presence of lipids or whatever, so we took vesicles coming from uninfected red cells, we couldn't see it. Only when we took vesicles and not something else, not just, you know, culture, you know, just media without the vesicles, for instance, when we took the vesicles, that was very strong to see the effect. So I think it's there. And the proteomic analysis, you know, showed us that the proteasome is there. So then taking these two pieces of information together really convinced me that this is something that's really specific. In terms of, you know, in vivo, I can't tell for sure because we don't work with, you know, that's one of the limitations with malaria. Most of our work is in vitro. We work with parasites that are growing in vitro and introduce them to vesicles, but we introduce them to lower amounts of vesicles. If we compare to, you know, the data that comes from clinical studies about the concentration of the vesicles from malaria patients, and even in your study, we introduced them to even way less vesicles. So in patients, there are way more vesicles that are released. So I think we are kind of in this range that, you know, from one side, we can't do the clinical studies, but we don't work, you know, we don't add more vesicles to see the effect. The effect is there, it's very strong, even with the low amount of the vesicles. And we also were able to identify the key proteins that the proteasome degrades, and that was very clean. So I think, you know, altogether, it means that there is something there.
Yeah, for sure. Thank you so much, Neta. Okay.
Thank you. Antonio, Marcela, please.
Okay.
Here you are.
Hi, Neta. Hi, everyone. Hi. My question, nice talk. I really enjoyed it. And my question related with the patient's samples. Did you check the presence of this whole proteasome in the patient sample? Is there? I mean, the parasite proteasome is already in the sample.
We look in the published data on, you know, proteomics from patients. We could detect proteasome there, but the problem is, keep in mind that when you harvest vesicles from patients, you harvest pool of vesicles, not just the parasitic vesicles. You get everything there, you know, it's from T cells, whatever is in the blood, you harvest the pool of vesicles. And now we don't have a good way to distinguish or to separate only the parasitic vesicles from this entire pool. So if you look at the pool, you can detect proteasome subunits there, but I can tell you for sure that this comes from the parasites, you know, secreted derived vesicles. But I think that the proteasome story is relevant to other systems as well, because we went over of many other databases in this proteomic Wikipedia, I can't remember the name, and we could see that in other systems from cancer and I think it was plants and like really diverse and other parasites, we could see that there are some proteasome subunits there. So I think it's more of general and it's not just for the parasites.
I agree. Thank you.
Thank you, George.
Hi, Neta. Fantastic talk.
Hi, Joe. Thank you.
Nice to see you, even though it's on the computer screen. Look, it's fantastic what you've done. And just a question about another cell type, which has been shown to be important in the destruction of malaria parasites. You know, Tony Ferrante here in Adelaide in Australia has shown that the neutrophils can be an important cell in the defense against falciparum malaria. So I know the neutrophils cannot synthesize proteins the same way a monocyte can do, but do you think there could be also a role for falciparum-derived EV on neutrophils?
Well, to be honest, we didn't look into that because it's a bit tricky with neutrophils, and I mean it is very fascinating direction. But once when we were thinking which kind of target cells to work with, neutrophils were a bit tricky. But I would say, yeah, maybe they have. If we have the system, it's something that we can easily test. But I would guess that there is something there, but I can't tell. We didn't look into that.
Okay. Thank you. Thank you. Congratulations.
Thank you. So Melissa Guadron-Lopez.
Okay. Hi, Neta. Thank you very much for this very, very nice talk. So I have two questions. One is regarding in how, can you comment a little bit in how can be the selective mechanism that lead to the cargo packaging if in these vesicles that manage to have nucleic acids, specific proteins, and now a full assembled proteasome inside the vesicles. How do you imagine that this occur?
You know, Melissa, this is the question that we get from the reviewers all the time. How can we explain that this cargo is selected into the vesicles? So the short answer is that I don't know. And this is, you know, we know from other systems that there are proteins that are involved in loading specific RNAs into vesicles or DNA or specific proteins. But in malaria, we are still not there. And I really hope that the field would go to this direction to try to identify the key proteins that are involved in cargo loading and sorting into the vesicles. But we are not there just because, you know, limitation of the system, CRISPR, or all of these genetic tools are not as easy in malaria. So we are limited. That's something we really want to do, and I hope that we will do it within the few more years. But we are not there. We don't know. We know that it's selective. I mean, we guess it's selective, a similar system of biogenesis of extracellular vesicles, but we don't know what are the essential components for loading and selecting these cargoes. We don't know. And even, you know, we even look at the cargo of the vesicles that are released through all the 48 hours life cycle. So every 12 hours, we harvested the vesicles and look at their cargo in terms of the DNA, for instance, or the proteins. And it's different. It's not the same. So at every stage of the parasite, there are other components that are released. For instance, the DNA is released only at the beginning, post-invasion into the red blood cell, the first 12 hours, and not later on. So there is no, at the trough stage, for instance, if you test the vesicles, you don't see a DNA, while you see DNA in the ring stage. So it is, as Jan said, it is very complicated, but at the same time, fascinating.
Exactly, exactly. Thank you for your answer. And the second question that I have is, do you have any evidence that the proteasome-dependent degradation of this cytoskeleton protein also might depend on polyubiquitination, right? To
Yeah.
label the proteins to the degradation in the proteasome. So do you think that the machinery that performs this polyubiquitination is also inside the vesicles, or is a signal that is in the host cell?
So it's a very good question. So according to the proteasome expert, Michal, so she, you know, I learned this field from her. So in many cases, you don't need this signature of the ubiquitin. So you need specific region that would be recognized by the core of the 20S proteasome, and then they start to degrade. So this is how we came to these proteins, because when we did phosphoproteomic analysis, we could see that these proteins, only when we introduced the parasitic vesicles, we got phosphorylation in regions that Michal said, these regions can be, prime them to be the substrates of the proteasome, just because of their structure, not because of ubiquitin. And indeed, we tested some of them, and these 4 that I showed you were the good examples of being degraded post-phosphorylation. So first, they need to be a phosphorylation event in this region, and then the exported proteasome degrades them. If you block the phosphorylation, there is no degradation. So it's more related to other post-translational modifications rather than the ubiquitin.
Okay, well, thank you very much.
So before we go to Maria, I would like to ask you, Neta, how far you've come into exploring which subpopulations of vesicles contain the different types of cargoes that you identify.
Yeah, so
it's not necessarily one type of vesicle, right? It could be multiple types.
Right, exactly. So we got this FFF apparatus here, where you can separate the different fractions of the vesicles. My student worked really, really hard to calibrate this system. And the system currently in our hands is very tricky. So every time we get a little bit different results, and it's not like we can see exactly the same populations of vesicles. So we are still not there. We know that there are several subpopulations, but so far it's from, I don't see that this FFF system give us like really, I don't trust it so far. I want to see that every time we do the experiments, we see exactly the same subpopulations, and we are not there. Maybe it's due to technical issues that we have with this system. It's very tricky for us, and we keep talking to the company, but we are not there.
So could you be looking at different surface molecules and identify subpopulation by vesicle surface?
So the thing is with the malaria vesicles is that we don't have the same markers as you have with the mammalian system. For instance, no CD36 or 9. So it's not the same, it's different. And the red blood cells don't contain all these markers as well. So we tried it a bit and it wasn't, we have one
Don't
marker.
give up, don't give up.
We'll keep working on that. I keep telling the students that once we calibrate this FFF apparatus, it would be good to get these subpopulations. We are not there. It takes us a lot of time.
I believe more in the surface molecules, but that's fine.
Really?
Maria Kuipers, please, can you unmute yourself and ask your question?
Yes, I can. So thank you, Neta, for your wonderful presentation. I got really exciting. So I was wondering, because the PVVAX is actually less severe than falciparum, I was wondering whether you know if they also use this CXCL10 as a sensing mechanism, just like the falciparum.
Yes, so I'm not the expert of Vivax. Hernando is the expert, so we can ask him. Vivax, it's a very, very tricky system to work. You can't grow them in vitro. So all the work that you can do is only with patient samples. And Hernando published an amazing story about these vesicles. I don't know about IP10. I mean, we didn't look into that. Maybe we should look if there is any, I'm not sure that if in the case of cerebral malaria, we see IP10, we will focus on Plasmodium falciparum, but that's something we can look into. If there are some, I don't know if Fernando knows, but or George, if there are some studies about that. If there are, we will look into that and see if it's similar. It's a good question.
And also because I think maybe it's more like a comment than a question, but I also read some papers on Leishmania also activating HER to modulate the host immune responses, especially I think it was cytokines in macrophages. And I think it's really interesting to see that more intracellular pathogens use this pathway. So I think you're just really on the tip of the iceberg there, like how this is modulated by pathogens.
Exactly. Yeah, I agree with you. I agree with you. And in our system, we took vesicles from trypanosome from Anna from Brazil, and we didn't see the effect. Like IP10 was getting secreted. There is no all this translation inhibition effect. So that was for us even, more exciting to see that maybe it is sort of specific to certain pathogens and not all the pathogens. But so as you said, the tip of the iceberg.
Yes, cool. Thank you.
Thank you.
Thank you. So if there are any more questions, please put your question into the notify me by putting something into the chat. Unless there's nothing else, I would say thank you very much, Neta, for giving us a lot of time for discussion. The length of your presentation was superb to allow that and keep us all alert. So thank you.
Thank you. Thank you for inviting me.
So, and the other thing I want to mention before I give back to Carolina is that most of you are aware that we let you in only if you type your whole name into the admission to the Zoom meeting. So just remember that you have to type your whole name and remind that to other people as well. So thank you very much. And I give the word back to Carolina.
Thank you so much, Jan, for reading the questions. And Neta, it was a wonderful talk. I was actually blown away. I was like, amazing result. And there's a lot of things to learn that didn't really show in the title of your talk, such as the ribosomal delivery. I think that is like a lot of people will miss out about that story. So I hope like the YouTube will help to deliver that story as well. So yeah, thanks so much for that. That was great.